So I'd like to go over today a little bit about some of the applications that you can do using CompuCell, some of the ideas. And then I'd like to come back and quickly, for people who want to stick around, cover one or two of the things that we didn't get through in the first module. As always, I need to remind you that the meeting is being live streamed and recorded on YouTube, and I'd like to thank our funding agencies for their support. We're going to be talking about simulations and some of the examples of things that you can do with them, help people think about the kinds of problems that you can address. You can see a number of them here on the screen. We're going to talk a little bit about the context for these kinds of simulations. And we heard quite a bit about this in the previous talk from Giulio. And then we'll go through quickly a couple of different examples, which might be of interest to different people in the audience. The modeling methodology we're talking about actually goes back about 30 years. And it's been used in a whole variety of different applications. Building this kind of model still takes some effort. To really build and publish a serious model takes quite a bit of time. Building simple models and being able to explore happens quickly. Building very sophisticated ones takes longer. Depends a little bit on what you want to do. And we can talk about that later on, especially if you go through the hackathon. Why would we want to build models of this kind at all? Well, we might want to understand the basic biology of the phenomena of development, maintenance, or disease. We might want to be able to predict the effects of chemicals, either toxins or drugs that we're using to treat diseases. We might want to be able to design new therapies. We might want to be able to personalize therapies for individuals. This kind of modeling is used in some situations to do personalized treatment. Some of the questions that we've worked with in our group over the years, and I'll show you some pictures, could be, why do blood vessels sometimes invade the retina as people age, causing blindness? Could we optimize treatments for individuals with diabetic retinopathy? Why do some people have uncontrolled growth of their kidneys, called polycystic kidney disease, that leads to death? And is there something we could do about it? Why does acetaminophen, paracetamol if you're in Europe, sometimes cause fatal liver disease? Or why does a drug affect or control or not control the spread of infection in a tissue? Some of the recent papers published using CompuCell specifically are on our website. There are more from the date of this screenshot. And I hope that the people who are participating in this workshop will generate yet more examples of CompuCell applications. You could see, if we look here, everything from ovarian cancer spheroids to radiation effects to modeling myxobacteria and influenza infection. So there's a wide variety of applications one can use this kind of modeling to address. Cancer modeling is one of the areas where the most has been done with multicellular virtual tissues. CompuCell is used, as well as Physicell and many other packages, to do this. One of the old simulations, which you've seen many times in the cover stories here of these slide decks, is trying to understand the interactions of blood vessels with tumors. We know that the tumors require nutrients and oxygen. Those are supplied by the vasculature. When those are insufficient, the tumors send signals causing blood vessels to proliferate, and those new blood vessels then interact with the tumor. There's actually a demo in the CompuCell demos folder that has a simplified version of this simulation, and we can try to understand that. We could also ask questions about how tumor evolution works, and we'll talk about that in a second. This is a simulation done by a graduate student in those days, Abbas Shirinifard, who's now at St. Jude Children's Hospital in Memphis, modeling a tumor spheroid. The tumor grows. the center of the tumor becomes hypoxic, doesn't have enough oxygen. It sends out growth factors, which then cause proliferation of blood vessels that are in purple. Those blood vessels grow and supply additional nutrients to the tumor. On the left, you see a simulation in which blood vessel proliferation happens normally. On the right, you see a simulation in which we blocked blood vessel proliferation with an anti-angiogenic simulated therapy. In both cases, the tumor grows. In the left-hand case, it grows faster. In the right-hand case, it moves further. We can compare the growth in these cases. This is tumor volume versus time. In both cases, as I say, the tumor grows, but the pattern of growth is different and the domains of growth are different. And so these are the kinds of questions we might begin to address and think about asking if we want to understand what's the meaning of our simulation. Gilberto Tomas, who's going to be teaching today, and others asked a question about what is called somatic evolution in tumors. We know that to become a frank cancer, you need to have a variety of things happen in the tumor. And these things happen in a predictable way. The cells stop dying when they should. They become resistant to immune killing. They don't respect normal senescence signals. They begin to produce their own growth factors. This list of what are called the hallmarks of cancer is quite interesting because from an evolutionary mutational perspective, mutations could be turning these factors up or down. So mutations, genomic mutations by themselves are not sufficient to explain cancer. Cancer actually is a disease of selection of mutation rather than generation of mutation, which is often actually not understood in some of the cancer literature. These days, microenvironmental or cancerous ecosystems become much more prevalent, but that wasn't the case even 10 years ago. And you'll be running a little simulation tomorrow, which explores some of this. In this simulation, it's a very simplified simulation. You have cancer cells proliferating. These proliferating cells differ in their availability of nutrient. When the cells divide, they have a small chance of changing the way they adhere to the extracellular environment or to each other. And what happens is there's an arms race where cells that adhere less to each other and more to the environment go to the surface and have more nutrient and therefore replicate more. And the result is that those cells can reproduce more. In this particular simulation, however, there's also a simulation of immune response. The immune response kills cells preferentially on the surface of the tumor. And so cells actually want to be or reproduce most, I don't want to give them intent, reproduce most effectively if they're just inside the surface of the tumor. The result of that is that this tumor stays compact. You'll see occasional cells separating from the tumor, and those are killed off by the immune system. On the other hand, if you have a weakening of the immune system or immune hiding by the cancer cells, then you'll see that in fact, the adhesivity between the cancer cells decreases with time, and the tumor will gradually spread out and metastasize into the environment. There's a question about, can CC3D simulate the emergence of cancer cells, or do they have to be hard-coded? The answer is a question of how long you're willing to wait and what kind of mutations you're willing to undertake. But yes, you can simulate, for example, this is a simulation of the transition between a benign non-cancerous tumor and a cancerous tumor. You could, in fact, simulate the evolution of tumorigenesis as well, if you were in the context, for example, of a normal homeostasis and a stem cell niche maintenance. And that's been done in the context of colonic crypt cells, for example. The main result here is that you see predictable pattern of a decrease in cell-cell adhesion and an increase of cell ECM adhesion, which leads to increased cancer invasiveness. And this is something that one could do quite easily in the level of this course. It's not a very complicated model, and it would be quite interesting to model the significance of this. One example of this, for example, is that it's known that in many solid tumors, treating the tumor by resection, by surgery, or by radiation or chemotherapy can actually cause the tumor to become more aggressive. That's because the tumor will be in a rough dynamic equilibrium with its environment. The rate of cell replication is limited by the nutrient availability. When you kill off large parts of the tumor cells can proliferate rapidly and evolve rapidly, and that can actually lead to the paradoxical outcome that treatment actually makes the cancer worse. In the context, a question about can we simulate growth of cells forming metastasis inside a hydrogel with specific properties? In principle, you can do that. You have to decide what aspect of growth and what aspect of the hydrogel you're trying to simulate. The question is always how you translate the experimental situation into the simulation. You can't model everything in infinite detail, and so the question is what aspects of that environment you want to replicate in the simulation. In the context of developmental biology, Pedro, who's going to be giving quite a few of the lectures in this class, has been interested in collective migration, which is important in wound healing, metastasis, and other situations. He was interested in trying to understand the mechanisms which lead to different patterns of migration in biological situations. These are all simulations of, sorry about the movie not playing exactly the way it should. These are all simulations of collective migration using different kinds of interaction between the cells. The top one is what's called the Boyd's simulation, where the cells move with a persistent motion by themselves and collide with each other. And that leads to one pattern of collective migration. The second one, the cells are responding to an external chemical cue and changing their environment. That's called self-generated gradient. You'll do that second simulation in this class. The third one, the cells have compartments with a leading edge and adhere to each other. You'll also do that simulation. The fourth one is a three-dimensional version of that same simulation. All of these represent situations which do occur in real biology. You've seen this segmentation movie before. It's sort of the prototype for a CompuCell simulation. In this case, you have a rather elaborate gene regulatory network and signaling network inside every cell that leads to oscillations. There is coupling of those networks between the cells through delta notch signaling, which you'll learn about on Monday. There's also a differentiation network that determines what the cells do depending on the state of that gene oscillation and in response to extracellular signals. And the cells then differentiate, change their adhesivity, and reorganize in response. And this is a simulation that was done by Giulio, again, Belmonte. We discovered in collaboration with the group of Claudio Stern at University College London that surprisingly, you could have somitogenesis in the absence of those chemical oscillations, those gene regulatory oscillations that were thought to be essential. And Giulio built a beautiful biomechanical model of that self-organization, which you might hear about next week in the advanced portion of the course using links and polarization. This is quite interesting because it shows that you can actually get segmentation through a whole variety of different mechanisms. Priyam Adhyapok, who's going to talk next week, built this simulation of segmentation as well in an epithelial sheet. And so one can explore both the biochemical control mechanisms and the biomechanical mechanisms that lead to these complex patterning in development. Another thing that Julio has done that he might be willing to talk about next week a little bit is build models of what's called planar polarity. These are signals within cells that give rise to gradients inside the cell. CompuCell can simulate these in a quite beautiful way. This is his simulation of the patterning of hair cells in the developing wing disc in a fruit fly embryo. Here you see the little vectors represent the degree and direction of polarization of each cell, planar polarity of each cell. This is a way of thinking about how to analyze these kinds of movies. We've worked quite a bit on retinal vascular diseases using CompuCell. The retina has a complex architecture, the black pigmented retinal epithelial layer at the back of the retina, the photoreceptors above it, and then neuronal tissue above that. It has a very high metabolic rate, and most of the nutrients and oxygen are supplied from outside the retina by what's called the choriocapillaris, which is separated from the retina by a thick membrane called Bruch's membrane. In some individuals, in particular in the elderly, but also in young people who get eye infections, the blood vessels in the back of the retina can invade the retina. And once they're there, they spread and they can actually destroy the mechanical integrity of the retina, leading to blindness. This is a simulation by Abbas Shirinafard, simulating how that invasion takes place. In this case, we were careful to calibrate the time scale over which the invasion happens. You'll notice this is a simulated year of time, focusing on adhesion defects, and in the bottom left, trying to understand how particular patterns of adhesion defects, a weak RPE POS, that would be weak adhesion between the pigmented retinal epithelium and the photoreceptors, would lead to retinal detachment after trauma. If you bang your head, you would have that situation. Weak basement membrane RPE, that would be to the front in that simulation, would represent reduced attachment to the basement membrane during aging, normal aging. And weak RPE would be what happens when you have an infection in the eye. Each one of those leads to different patterns of damage in the retina, and those can be then seen clinically. And that suggests particular approaches to therapy, which were actually validated, in this case, in experiments after that original publication. We've worked on trying to improve therapies for diabetic retinopathy. In this case, one of the therapies, not used as much today as it was when we did this work, is to go in with the laser and actually destroy parts of the retina that are causing problems. The problem is this is like firefighting or forest fire firefighting, where you cut down trees to make fire breaks. The problem was that people couldn't predict where to build the fire breaks. And since you're actually destroying parts of the retina, the treatment is pretty harsh. There's a model of how this damage works. Hypoxia in the retina leads to damage, cascade of damage of the blood vessels. That's very much like a forest fire model. And you can build a simulation based on actual images of patient vasculature in the retina of what the probability, if a blood vessel fails, what's the probability that that will cause additional blood vessels to fail. When a blood vessel fails, it causes hypoxia. That causes other vessels to be more likely to fail. Given that, you can ask the question, if you were going to kill cells in the retina, which ones should you kill to cause the minimum area of damage from the killing and do the maximum you can to prevent spread of propagating vascular failure? And you see on the right a trade-off matrix where if you do less treatment intervention, you cause less direct damage, but you have a higher probability of the disease progressing and vice versa. Polycystic kidney disease, as I mentioned, is a disease in which the kidneys continue to have cell proliferation, not in normal architecture, but in the formation of cysts. It's a genetically caused disease. If you have a mutation in polycystin 1 or 2, you're almost certainly going to get it. If you don't, you're almost certainly not going to get it. But we didn't understand how those mutations related to the phenotype of the disease. Again, Giulio was very much involved with this. In this case, as before, we focused on cadherins, the molecules that help cells adhere to each other. In this case, there was an experiment that showed that in cells which were going to be part of a cyst, there was this cadherin present, a cell adhesion molecule present, that was present in children, but not in adults normally. In vitro experiments, you introduce that molecule into cells, and they immediately form cysts, and only cells with that molecule expressed form cysts. However, adhesion molecules do multiple things. They cause cells to adhere to each other, but they also contribute to cell signaling. And Giulio designed a set of simulation experiments in which you interfered with cell signaling property of the cadherins or the cell adhesion property of the cadherins. When you run those simulations, A, you're interfering with, and in the movie, you're interfering with adhesion. B, you're leaving adhesion alone, but interfering with signaling. In both cases, you get cysts, but the rate of growth and the pattern of growth is different. And in the experiment, you can see that the pattern of growth corresponds to the decreased adhesion case, much better than to the decreased contact inhibition case. In vivo, in patients, there's actually a spectrum of these, and you probably have patients in which one factor is dominant, and some in which the other factor is dominant. But this was a response to the question of how you decide what's meaningful of a simulation. In this case, the time course of evolution of the cysts is important. The spatial organization of the cysts is important. They're very different for different hypotheses about the mechanism of the disease. That led to some classic bioinformatics work, and then to some drug targets and drug candidates that are now being tested in animal models to see whether we can actually come up with a drug therapy for this disease. At the moment, the only treatment is a kidney transplant. We've worked a bit on infectious diseases. This is a simulation that we actually started out as a class project in a class containing mostly undergraduates back in 2020. In March 2020, the students said, instead of studying cancer this semester, can we study infection in the lung? Do a little simulation of COVID or influenza. The students built a simple model in class, worked on it with some other people over the summer, and it was published the following year in PLOS Computational Biology. In this case, we started out with a single infected cell and a patch of simulated epithelial tissue. The infection spreads through the tissue by replication and release of the virus in the cells. That replication and release sends signals, cytokines, which call in immune cells, and the immune cells then kill infected cells. In this simulation, the immune cells can't keep up and all of the cells die. But you can study, for example, the effect of different kinds of treatment, when you give the treatment, how much treatment you have, which aspects of the viral replication you target, how effective the treatment is, and come up with approaches to trying to quantify how effective treatment strategies would be. There's a huge amount of variability individual to individual, and that's something you can also study in this kind of simulation. One of the things that we wondered about was how often you'd have to treat with a particular drug. This is a simulation of remdesivir that Giuliano, who's going to be helping with this class and giving some lectures, worked on in his PhD work. And he was particularly interested if the cells didn't metabolize remdesivir in the same way. Remdesivir is a prodrug. It has to be turned into an active form inside of cells. If the rate of that metabolism varied between cells, what would happen? And the result in his simulations, which were published last year, is that higher rates of variability mean you have to give much higher doses of the drug to be effective. That was a fairly simplified model of the immune response. T.J. Segoe and others in collaboration with the group at Pitt and others in Pittsburgh built a simulation that was validated against very detailed animal experiments of influenza, modeling a whole variety of immune cell signals and immune cell types in the lung. This is still not the full complexity of immune response in the lung, but it's getting pretty close. We have quite a few of the main cell types. We have a reasonable representation of how immune cells are generated in the lymph nodes. And a model like this could really be turned into, I think, pretty easily, quite a reasonable model of the actual pattern of immune response in the lung. Another area that I mentioned that we've worked with is computational toxicology. We were interested particularly in hepatotoxicity, liver toxicity, due to Tylenol, acetaminophen. In this case, we actually have an explicit model of flow in the liver. In this case, we're looking at how the pattern of flow in the liver delivers the drug to different liver cells. Here, we're modeling a single sinusoid, a single capillary in the liver, and looking at the damage pattern caused by the chemical that's carried in the flowing blood to the various cells in the liver. And this we're able to then calibrate against experimental measurements, or clinical, in this case, clinical measurements of the level of acetaminophen in the bloodstream in an individual patient and the level of its metabolites. People have used the CompuCell in a whole variety of computational toxicology studies, mainly people at the U.S. Environmental Protection Agency. As always, when I say that, I have to say that these are not the opinions of the EPA, but they've studied vascular disruption in embryos. They've studied urogenital development in embryo, disruption in embryos by estrogen mimics, and disruption of the formation of the roof of the mouth, cleft palate, in embryos as well. Last year, we had Shane Hudson come and talk about that simulation, present it to the group. I think quite interesting. There was some beautiful work on liver zonation, more detailed work building on what we just showed on the Tylenol study, with very detailed models of how the liver processes chemicals. And that was actually work that was presented last year by a high school student. So these things can be quite interesting when you have a chance. That was what I wanted to talk about methodologies. I did want to quickly walk through some of the material, and I know we'll be a little bit over time. I've asked you to give me 10 or 15 minutes of your time. People are free to leave and go get a bite of lunch, especially if you're not NanoHub people. But I wanted to talk a little bit about how to use Twedit and Wizard to create a cell growth simulation in NanoHub. And I'm going to do a reshare of the screen here. And if you want to fire up NanoHub and try to follow along, you're welcome. I'm not going to expect people to be able to do all of this, but I do want you to see it because you're going to be doing this repeatedly during the rest of the day and during the rest of the week. And I wanted to give people a once through here before we go forward. So I'd like people to take just a minute to either fire up your desktop CompuCell or your NanoHub CompuCell, either one. And I'll wait just a minute or two for people to do that. In the meantime, I'm going to start out with CompuCell. Actually, I had CompuCell running already. I didn't need to launch it again. Okay. And what I'd like people to do once you're in CompuCell is to start Twedit. And I'll do a quick poll. And now what I would like people to do is go to CC3D project. Here, and do new CC3D project. And this should work either in player, in NanoHub or desktop, shouldn't matter. And we're going to create a new simulation. And by default, it gives you the name new simulation. I have a lot of these, so I'll call it new simulation 10, just so I don't run into the simulation I've already run. But you can call it any name you like. There are a couple of constraints on the names that you use. Do not use a number or a special character at the beginning of a name. Don't put a space in it, and don't use special characters other than underscore in it. So be sensible in your naming conventions. Simulation _1 is fine. 7 exclamation point space 3 is a bad choice. Okay, and we'll hit next. If you have a problem with this, you may have to navigate. Let me go back a step. When we create that simulation to begin with, you may want to select hit browse here and make sure that you're pointing to your user directory on NanoHub. If you select as a target a directory that you don't have write access to, you'll get an error when you try to save the simulation. So I'm trying to write to something you can't write to. For simulation properties, we don't have to make any changes here. We're going to live with those. And so we'll just hit next. Okay. We're now going to specify the cell types, and we'll just call them cell 1. And we'll hit add. And we'll say cell 2, and we'll hit add. Let me nudge the screen over just a little bit so you can see better. So we have 2 cell types. So everybody should make sure you have 2 cell types. Then hit next. We're not going to have any chemical fields, so we hit next and skip that. And now we select our cell properties and behaviors. We're going to say the cells have a contact energy, they stick to each other, that they grow, that they divide, and then we have to say that they have a volume, because we don't get anything for free. Okay, everybody can do that, I hope. Contact, growth, mitosis, volume. Next. Configuration complete. Hit finish. And here's the simulation. We can save the simulation by hitting Save All here, which I recommend doing periodically. And now you want to open the simulation. So I'm going to open the simulation in Player by hitting the CompuCell icon. And it'll open Player. And I can hit Run. Oops, I've got the wrong simulation there. I want this one. I want to open this in Player. There we go. I have to joggle my screen to get a redraw. There we go. And you see I've created a number of initial cells here of the 2 types. And if I hit Run, those cell types will grow and divide and change. And you can play with changing the colors, changing a variety of aspects of that simulation. Remember to start, stop, step in the control panel of the. Okay, let me just walk you through in just 1 second what the simulation specification looks like. The first part of a simulation is in XML. And the second part of the simulation is in Python. And most simulations, not all, but most simulations will contain a mixture of those two languages. In general, the static aspects of the simulation are specified in the markup language. The dynamic aspects are specified in the Python. To see that, we double click in Twedit on the name of the simulation. It'll open up a cascade of windows, 3 in this case, that specify the simulation. If we look inside the simulation, at the beginning, we'll see a block called metadata that specifies whether to use parallelism in the simulation, whether we need debug outputs or not. And then a POTS block that specifies the size of the lattice of the simulation, the temperature fluctuations used, the amplitude of fluctuations, and the interaction range when you're doing lattice updates, which Julia talked about just before. The next block is, in there we see, for example, the dimension of the simulation is 256 by 256 by 1. It's a 2D simulation. I made it 256 by 256 by 256, be a 3D simulation. The next block of code specifies the list of cell types in the simulation. We have 2 cell types, cell 1 and cell 2, and we always have something called medium that specifies what happens where I don't have a cell. We then specify what are called plugins, which specify those energy functionals that Julia talked about. And those plugins are, for example, contact energy. What is the contact energy per unit area between 2 cells of different types? Here we have the contact energy between medium and cell 1, medium and cell 2, cell 1 and cell 2, cell 2 and cell 2, and so on. In this case, the default is that they all have the same energy. Everything has the same energy of contact. We can change that if we like. We could edit that as we run. The last thing we do is we specify an initial condition. In this case, Uniform Initializer draws a rectangle of cells of different types with specified sizes over a specified region. If we look at the Python, there are a couple of things we'll notice. We'll see something called the start function that's run once at the beginning of the simulation. In this case, we iterate, remembering yesterday, iterating over the list of all the cells and giving each cell a target volume, and it will target compressibility. You'll hear more about that later today. The other thing that we do in a simulation, which is quite simple, is that we have the cells grow. We have a time step. We go through each cell and we increase the volume of each cell by 1 at each time. That's probably too fast to be very useful, but for a game, it's okay. And the last thing that we do is that we have the cells divide. In this case, at every time step, we make a list where we store the cells that we might want to divide. If any cell is bigger than a volume of 50, we say to divide it. We then go through the list of cells we're going to divide. We allow them to divide in a variety of orientations, in this case, random orientation. And after the cells have divided, we have to say what attributes the daughter cells have. And there's an update attributes function that does that. In this case, we say that half of the volume of each parent cell, the parent cell goes to each child cell, and otherwise the child cells are the same. And then, just to make life complicated, if the parent was of type 1, make the daughter of type 2 and vice versa. And that's what gives us that simulation. And so now we can run that simulation again. and we can see what it does. Again, you'll see the colors will change. We'll see the cells grow. When they get to a certain size, they divide. And if you watch carefully, you'll see that the daughters have a different color from the parent. And so that is a simple first simulation, which you can run without changing and doing any coding at all, just from the controls. And I think the best way to get comfortable is to see these things multiple times. You'll go over this in much more detail later, but I did want to walk you through it now so you've seen it. Practice and is really essential in these kinds of simulations. Takes time to be comfortable with how CompuCell works, how Planar works, figuring out if there are bugs on your specific system. And so we'll cover the same things multiple times to try to help you be sure that you feel comfortable with all the steps involved. It says, if I missed out on checkboxes of general properties page, how do I add them back in? If you've closed Wizard and you have already set up the simulation, then you will have to open in Twedit explicitly the simulation and add them there. And Gilberto will show you how to do that today and tomorrow. If it's such a simple simulation as the one we did, you might just want to restart Wizard from the beginning and create a new simulation where you tick those boxes. If you haven't hit save a simulation at the end of Wizard, you can always hit the back button and redo it. But once you've wrapped the simulation, you have to start over. There's a good question from Peter. Does the 256 by 256 box start killing cells due to high pressure? Absolutely. This simulation, again, in this world, nothing exists unless we assert it. And we're living in a finite size domain. And these cells are growing at a fixed rate. In reality, cells, when they're under pressure, stop growing. And so one of the things that you'll learn how to do tomorrow is to include what's called contact inhibition, where if the cells are under pressure, their rate of growth slows down and stops. And if we included contact inhibition, in this case, the cells would fill out the domain and then they would stop growing. We'd have to introduce some kind of killing of the cells, representing normal cell death or something like the cell death due to chemotherapy or radiation to induce growth after that had stabilized. Great question. There's a comment about random seeds. Yes, it should be slightly different each time you run it. Let's see here what happens. Again, remember in NanoHub, pulling that corner really helps on screen redraws. Let me change my colors so they're a little bit easier to see the difference between the two cell types. So let me time step it. Okay, see the cells growing, dividing. It's maybe a little bit less obvious, but they're changing type. Let's zoom in. That's too much zoom. Let's zoom in a little bit. Boy, I have a new laptop. And the touchpad is not that friendly for this purpose. I'll just use this. There we go. There we go. So I can see the individual cells better. I'll time step that. You can see the cells grow. Let's see somebody divide. And you should see that if a yellow cell divides, I get a yellow and a green cell offspring. If a green cell divides, I get a yellow and a green cell as well. I'll let you explore that a little bit.